All right, I'm pleased to introduce a colleague of mine in radiation oncology, Zin Kai. Zin did undergraduate in chemistry and physics at Harvard. Already started to get a master's degree there when he was studying some biological aspects. He was accepted into the medical scientist training program here at UT Southwestern. After the first couple of years of medical school, he entered the lab of James Chen in molecular biology and studied the basic science and immunology of the innate pathways, as well as some impressive mechanisms for antiviral defense. And they were published in very good journals. He finished medical school and then was drawn to radiation oncology. And typical, he did his residency at a top-tier program, Memorial Sloan Kettering in New York City. Along with his training, he went back to the lab and worked in the lab of Craig Thompson, a very notable person there, where he made a pretty big risk, in my opinion. He completely changed his area of focus after having very successful start and started to study metabolism. And normally we think of metabolites very akin to food in a food bank or a pantry sitting around waiting to be consumed, but he took a very different slant on this, which he'll talk about. What I'm also impressed by Zin is that he's a passionate physician. He believes that studying questions in the lab based on clinical observations is a very useful tact. And he's the type of person in a clinical department that's really going to be transformative. So I'm really happy to introduce you.
Right. Thank you for that very kind introduction. So I want to share with you a little bit about how my lab and how I've thought about the field of metabolism. So these are all the metabolic pathways that we've learned and some of us have forgotten. But the question is, is this all metabolites do? Because this is essentially a map, right? That tells us where different metabolites go, the chemical reactions that they undertake. So this is almost like a map of Dallas. But what we care about the most is not only where they go, but what they actually do. So for example, on the left here are two very commonly used drugs. They are Tylenol and also metformin. And on the right are two metabolites that share some structural homology to the drugs on the left. For the molecules on the left, we ask what they bind to. That's what people care about. We want to know the target of metformin and the target of Tylenol. But for the things on the right, we care about what they turn into in the cell and how they can be used as fuel. So we want to understand essentially what metabolites do as small molecule drugs. And this really started with our clinical work as a radiation oncologist. I treat patients with a number of different solid tumors. And one of the scans we almost always get in any kind of cancer patient is something called an F18 FDG PET scan. And this is the principle of which dependent on the principle that cancer cells significantly upregulate their utilization and uptake of glucose, right? So this is essentially a tracer for how much glucose is taken up in the cell. And as you can see, the tumors here in the liver take up significant amount of glucose, and after treatment, they're resolved. So what is the principle behind the PET scan? It's actually something you've all heard of. It's called the Warburg effect. It's not 100% correct, but what essentially happens in a normal tissue is that the glucose can, go through glycolysis, gets turned into pyruvate. And this pyruvate and most of the energy production can happen in the mitochondria. But in tumors, they significantly upregulate their glycolysis such that the glucose amount in the tumor microenvironment is much lower than the normal tissue, and their lactate level builds up very significantly, right? The ability of human tumors to upregulate glucose uptake made us ask, what happens to the end product of glycolysis or lactate? So the two questions we started to ask were, are there any signaling functions of lactate that are independent of its metabolism? And as shown by Ralph and a number of other people, lactate can be used as a metabolite, a fuel source in the mitochondria. So how do cells know? How do cells sense that there's abundant lactate around so that they should use it. Just so to answer this question, we took a chemical approach. So lactate is a chiral molecule. L-lactate is what you and I produce. It can be metabolized by this LDH enzyme. D-lactate, the mirror image, is unable to be metabolized in most of our cells, right? So this is a non-metabolizable form of lactate. What we found by measuring the cellular's oxygen consumption rate in the mitochondria was that both L and D lactate are able to significantly increase the amount of oxygen that's consumed by the mitochondria. This was surprising because we know L-lactate maybe can be metabolized. I might explain this effect, but D-lactate is not metabolized. And surprisingly, when we culture cells in galactose such that the entire cell's ATP must be generated from the mitochondria, we found that a non-metabolizable form of lactate is significantly able to enhance cell proliferation, suggesting somehow in the presence of this molecule, the mitochondria becomes more efficient. So we then, having trained with James, we did a lot of in vitro assay. We then took a somewhat biochemical approach to ask whether or not there are signaling functions. And we took this assay, which was we used a cell-free mitochondrial assay to check the activation status of this PDH complex. This PDH complex is a very important enzyme complex that converts pyruvate into acetyl-CoA, which then enters a TCA cycle. The complex can be activated or deactivated through a series of phosphorylation by kinases and phosphatase. So it's inactive when it's phosphorylated, and it becomes active if it's not phosphorylated. And as you can see here, both L and D lactate in a purified mitochondrial system can suppress the phosphorylation of the PDH complex, meaning that it's becoming active, right? So this is done just with these metabolites with no enzymes, and there's no metabolic reaction happening. And dichloroacetate here is a kinase inhibitor, and calcium here is a phosphatase activator. Here are the controls. So next we asked, what could be the possible mechanism? The electron transport chain, as some of you know, is really dependent on the availability of oxygen as a terminal electron acceptor. In acute hypoxia or the absence of oxygen, the electron transport chain is not functional. And we found that the ability of lactate to activate the PDH complex is dependent on oxygen availability, such that acute hypoxia essentially blocked lactate signaling. Somehow this suggested to us that the mitochondrial electron transport chain may be important. So to do this in an intact cell, we ask whether or not cells containing mutations in the mitochondrial electron transport chain. So these mutations, they don't completely abolish ETC activity, but they compromise ETC efficiency. Whether D-lactate can enable these cells to grow and proliferate. And it turns out using a cytochrome B mutant patient-derived cybrid cell line, D-lactate can significantly enhance the proliferation of these cells. And similarly, when we use a low dose of rotenone and antamycin to block or to inhibit the electron transport chain, D-lactate is able to enable these cells to grow in the absence of pyruvate. So our take-home message is that we found that lactate can enter the mitochondrial matrix, which I didn't show you here. And after it enters the mitochondrial matrix, it can stimulate OXPHOS, which then leads to the activation of this PDH complex. This PDH complex activation leads to increased flux of pyruvate into the mitochondria, such that more lactate is then converted into pyruvate. So it's essentially a feedback cycle to increase this pathway while suppressing the glycolysis pathway. So we propose that lactate signals its own preferential use over glucose for TCA cycle, and the mitochondrial electron transport chain can sense the levels of lactate. So here are some of the ongoing projects in my lab. Metabolites are small molecules. They're made of carbon, nitrogen, oxygen, and hydrogen, just like any other drugs. They have no metabolic functions. But what we're interested in is to understand their functions independent of their metabolism, where they can serve as a drug, bind to a protein to either activate or inhibit the protein. We have ongoing project to look at the mechanism of lactate signaling to the mitochondria. We're looking at the regulation of cell fate decision or cell differentiation by a number of other cancer-associated metabolites, lactate being one of them. And lastly, we have an interest in looking at the functional metabolites derived from commensal bacteria, because unlike you and I, we use L amino acids and specific forms of glucose. Bacteria use both, right? And the D amino acids that are produced by the bacteria is not metabolized by us, but we feel like they must have important signaling functions. We use tissue culture and cell molecular biology. We use in vitro reconstitution and some biochemistry, and of course, mass spectrometry for both protein and metabolite quantification and identification. And here are the people in my lab and many of the collaborators here at UT Southwestern. Thank you.